Okay, I'm going to pick up here with the visual quiz 11.4, where we are labeling the phases of meiosis. Okay, so the one phase we're going to have just the one going into two cells. So if it has one cell like this one, and this one, interphase. This one's already labeled for you. Interphase. Remember, that is the phase of the cell cycle that's not actually part of mitosis or meiosis. But that's where the cell's doing a lot of growing, making proteins, and all that good stuff, getting ready for the cell division. Okay, we see right here that the centrioles are starting to move to the opposite ends of the cell, and the sister chromatids, the homologous chromosomes, in this case homologous chromosomes, are pairing up. So this would be prophase 1. So prophase 1. Okay. Prophase 1. And then metaphase is, meta means middle. So this would be the next one. They're lined up in the middle. Notice that they are tetrads. Tetrad means 4. Tetrads, you have these are the homologous chromosomes. One came from mom, one came from dad, and they match up. One came from mom, one came from dad, and they match up. So right here in this phase, we are matched up in tetrads, homologous chromosomes. They're lined up in the middle. The centrioles are already at the ends. They're starting to send out spindle fibers. This is metaphase 1. So meta 1. And then they start to pull away from each other. Ana, PMAT, prophase, metaphase, anaphase. Ana means away. So that right here they're starting to pull away from each other. So this would be anaphase 1. Anaphase 1. And then PMAT, prophase, metaphase, anaphase, telophase. Tele 2 starts splitting into 2. So this is one cell starting to split into 2. So this would be telophase 1. Okay. And in meiosis, we go through that process 2 times. So at the end of telophase 1, cytokinesis 1, we end up with 2 cells. And these are 2 cells with half as many chromosomes. Okay. But the chromosomes are still in X's. Remember, mitosis is X's split apart. Well, during the first round of meiosis, they stay together, so we still have those X's. And now both of those cells that we just made going through meiosis 1 are going to go into meiosis 2. And we already have early prophase labeled here, and that's just where and sometimes it doesn't even really happen because it doesn't always have time to even form that new membrane or anything. It just goes straight into metaphase and anaphase. Okay, so if this is prophase 2 early, this would be prophase 2 late. Okay, because we've got the centrioles, we have our homologous chromosomes together. Okay, I mean our little X's where it's condensed into the little X's. Okay, so this would be prophase. Meta means middle. So this time we have 2 cells with our chromosomes lined up down the middle. Okay. And again, this is like mitosis this time. We don't have tetrads when we go through the second part. We have the sister chromatids still attached at their centromeres here. So this would be meta means middle. So this is meta 2. And then those are going to start to pull apart. So ana means away. They're pulling away from each other. And this time the sister chromatids separate, like we saw in mitosis. This time the sister chromatids separate and pull to the opposite ends. So this is anaphase 2. And then telophase 2. Both of those chromosomes start to split into 2. So we end up with 4. So at the end of meiosis, we end up with 4 new cells, and those 4 cells are unique. None of them are just alike. When we pair up over here in prophase 1, we get crossing over. Part of this chromosome may break off and stick to that one. Part of that one break off and stick to that one. They trade some genetic material. So that gets some diversity right there. And then they can assort and separate different. We get independent assortment. So at the end of meiosis, we get 4 totally different chromosomes, or totally different cells, gametes in there. Remember, at mitosis, we got two cells that are identical at the end of mitosis. At the end of meiosis, we have 4 cells that are all unique. And that's where we get the genetic diversity that is so important to survival. Okay, so number 1 down here says, meiosis results in the formation of how many cells? Well, that would be 4. And they are haploid. They have half as many. Okay, haploid. They have half as many chromosomes as the body cells do. And that's because we're going to have eggs and sperm, and half your chromosomes going to come from the egg from the mama, and half the chromosomes are going to come from the sperm from daddy. So those are haploid. So number 2, which shows the correct sequence of events in reproduction? So we know we're going to have to start with gamete formation. So we get gametes. And then when the egg and sperm meet, that is called fertilization. Forms the zygote. So C would be our correct answer. Okay. Start with gamete formation. So we know that eliminated those 2. And we can't have a zygote before we have fertilization. The zygote is that back to that diploid stage where after the egg and sperm come together. That makes the zygote. Okay. So a human zygote contains 46 chromosomes. How many chromosomes does a zygote get from its mother? Well, if there's 46 total, then 23 came from the mother and 23 came from the father. Number 4 says, if a somatic cell of an organism has 30 chromosomes, remember somatic cell means body cells, and that's all the body cells. Somatic cell has 30 chromosomes, and the germ cell produced during meiosis would have how many? Well, the germ cells are going to be haploid. They're going to have half as many. So if the somatic cells have 30, germ cells are going to have 15. During which stage of meiosis does crossing over occur? Well, we just looked at it right up here. During prophase 1, when the tetrads are forming, those homologous chromosomes are pairing up, that is when you're going to get crossing over. So that's going to be prophase 1. Okay. So crossing over results in new genetic combinations, and that's where we get genetic diversity that is so important to survival of a species. Okay, number 7, what is a tetrad? So a tetrad is when homologous chromosomes each with 2 sister chromatids pair up. Okay, and what that looks like is you would have chromosome number 1 from mom and chromosome number 1 from dad, and then during interphase, remember those chromosomes replicate. So this is just a copy of this one, and they're held together right there at the centromere. It's still counted as one chromosome because you count the centromeres. It's the same thing. It's just copied getting ready to divide. And then the one over here from mom, it also copied itself. So it has 2 sister chromatids. So this is 2 chromatids, and this is 2 chromatids. These are homologous chromosomes, and during prophase, they pair up together. So we end up 1, 2, 3, 4 sister chromatids, and it's called a tetrad. And then when they're paired up like that, that's when you can get crossing over, and it doesn't cause an issue. Because if you take this part off of this one and attach it right there and swap that one over to there, then they're homologous, so they have the same genes in the same place, so that doesn't cause a problem. Okay, that happens with those homologous chromosomes during prophase 1. So what is the process called that forms a tetrad? Well, to form the tetrad, that would be, first you would get DNA replication. and then pairing of homologous chromosomes. During which stage of meiosis does this process occur? The forming of the tetrads. Again, that happens during prophase 1. And number 8 says, at the end of meiosis 2, how does the DNA in each of the new cells compare to the DNA of the original cell? Half as much, that's correct. So the two that say half as much, we can eliminate the ones that say twice as much, because that's not going to happen. They're going to be haploids. They have half as much. Genetically the same or genetically different? Well, we said we get crossing over that makes them different. We get independent assortment of genes, so they are going to be half as much genetic material and genetically different from the original. So you get 4 haploid, genetically diverse cells. Okay, we're comparing meiosis and mitosis. And we're going to say, is this part of mitosis or is this part of meiosis? Number 1, no pairing of homologous occur. Now, it does occur in meiosis. It does not occur in mitosis. 2 divisions. Meiosis goes through PMAT1 and PMAT2, so meiosis has 2 divisions. 4 daughter cells are produced. Well, we only get 2 in mitosis. So we get 4 in meiosis. Associated with growth and asexual reproduction. Well, if it's asexual, then that's going to be mitosis. Meiosis is forming gametes for sexual reproduction. Associated with sexual reproduction, well, that's going to be meiosis. 1 division. Mitosis only goes through 1 division. Meiosis goes through 2. Mitosis goes through 1. So that goes under mitosis. 2 daughter cells are produced. Well, because mitosis goes through 1 division and forms only 2 cells, and they are genetically identical in mitosis. Daughter cells are produced. Okay, both of them. The daughter cells are produced in both. Chromosome number is maintained. That means it has the same number. Well, in mitosis, it's maintained. We have the DNA replication, the sister chromatids separate in that 1 division. In mitosis, we separate our homologous chromosomes, which gives them half as many chromosomes, and then sister chromatids separate. Chromosome number is halved. Again, that's meiosis. Crossing over between homologous chromosomes may occur. That's meiosis. We don't have homologous chromosomes in mitosis. Daughter cells are identical to the parent cell. Mitosis, we get 2 identical daughter cells. Meiosis, we get 4 unique. Chromosome number is number 13. Daughter cells are not identical to the parent cell. That's meiosis. They are all unique. Produces gametes. That's meiosis. And forms a tetrad. It occurs in prophase. So that would be meiosis. So that's what that should look like.